what I will show you today is some of our results that will hopefully lead to the identification and possibly to the clinical development of novel candidate molecules for the treatment of myocardial ischemia and heart failure. So this is an X-ray showing the coronary arteries, so the vessels of the heart of a patient with a severe myocardial ischemia. You see here there are several stenosis and a poor distal perfusion of the heart. And the thing is that at present we cannot do much for such a patient who is destined to inevitably evolve toward a heart failure phase of his disease. And this is why we need to develop novel strategies to induce the formation of new blood vessels in this heart, so to induce a neoangiogenesis process, to protect cardiomyocytes from cell death, and possibly to induce myocardial regeneration. And different tools and approaches have been proposed so far to achieve these ambitious goals, and we don't have time to enter into details, but we can reasonably say that the use of both recombinant proteins and cell therapy using several sources of putative stem cells have already reached the clinical phase, and they eventually gave negative results. Whereas gene therapy might be more promising in this sense. And among the different systems that we have at present to transfer genes into the heart, the most promising one is for sure the one based on the use of vectors derived from the Adeno-Associated virus. And this is because this virus is very diffuse and it's not pathogenic. So 80% of us do have antibodies against AAV, meaning that we have been in contact with the virus. The recombinant vectors don't express any viral proteins, and this means that these vectors are not inflammatory nor immunogenic, thereby allowing long-term expression of the transgenes in vivo. And finally, these vectors have a specific tropism, so they work very well in all kinds of postmitotic tissues, such as the skeletal muscle, the heart, neurons in the brain, and different kinds of cells in the retina. We don't have time to enter into the details of these specific tropisms, but if you're interested, I invite you to go to the posters of Miguel Mano and Yasmina Lovric and interact with them about this topic. So, starting from the consideration that in regenerative medicine, whatever tissue you want to regenerate, you will almost always need to create new blood vessels to support oxygen and nutrients to the regenerating cells and the newly forming tissue. Our story started with the idea of exploiting AAV vectors to overexpress angiogenic factors and to induce new blood vessel formation into ischemic tissues. And as you may know, angiogenesis is a very complex process requiring the coordinated action of several growth factors, cytokines, enzymes. But for sure, one of the major players and the most important players is VEGF, the vascular endothelial growth factor, which per se is able to induce the proliferation and the migration of endothelial cells from a pre-existing vessels, thus driving the formation of new capillary sprouts that subsequently have to mature to form functionally competent arteries and veins of different sizes. And the angiogenic activity of VEGF-A, and in particular of its most potent isoform, which is composed of 165 amino acids, is essentially mediated by its binding to VEGF receptor 2, which is abundantly expressed on the surface of endothelial cells. So what happens if you take an AAV vector to overexpress VEGF-A and you inject it either in the skeletal muscle or in the heart of an animal? This is the answer after 1 month, and this is a staining for an endothelial cell marker. So these small circles are the capillaries normally present in a skeletal muscle, whereas this is after 1 or 3 months of VEGF overexpression. You can see here there is a huge increase in the number of the capillaries. This is another way at looking at the vessels. In this case, we have in vivo perfused the animals with fluorescinated lectin, and the lectin binds to the endothelial internal layer of the vessel, so it stains the internal layer of the vascular wall, and this is the normal pattern of capillaries surrounding each muscle fiber. This is a higher magnification, whereas this is after VEGF overexpression. So from this experiment you can also appreciate that the vessels formed by VEGF-A are very leaky, as the lectin has the tendency to extravasate from the vessel lumen and permeate the tissue. But besides doing new capillaries, VEGF is also able to form arterial vessels. Here again you have an immunostaining for alpha smooth muscle actin, which stains this smooth muscle layer of the arterial vessels, and this is the normal number of arteries present in a muscle, whereas this is after 1 or 3 months of VEGF overexpression. Again, we cannot spend much time in explaining how these vessels reach their functional and structural maturation, but there are 2 other posters by Alessandro Carrera and Marian Casemi that better explain this topic. And of course, thinking at going toward the clinic with this angiogenic strategy, we wanted to move to a larger animal model. And at the New York Medical School in Valhalla, they have a very nice model of myocardial infarction in the chronically instrumented dog. So basically what they do at time 0, they insert into the coronary artery of the animal an hydraulic occluder, which will allow to induce a myocardial infarction. And then they place several pairs of piezoelectric crystal throughout the heart. And this crystal will allow to monitor local cardiac contractility by echocardiography. And then they also place a series of other transducers to monitor blood pressure and flow over time with living animals. So in this system, what we did is, after 14 days from the animal instrumentation, we induced the myocardial infarction by inflating the hydraulic occluder. And after 4 hours, and we decided to choose this time point trying to mimic a possible clinical scenario, we injected 2 AAV vectors coding for either VEGF-A or LAC-Z as a control. And this is the result. This is not the result. Okay, this is the result. So by using the piezoelectric crystal, as I told you before, you can monitor the cardiac contractility in a very localized manner. So if you look at the contractility of the infarcted region, you can appreciate this negative curve, which means that while the whole heart is contracting during systole, the infarcted region is not able to contract and it bulges out. And this produces this negative curve, which is progressively reabsorbed as the fibrotic scar is forming until that region becomes completely akinetic and cannot move anymore. And this normally happens after 1 month. Instead, you see that in the animals that had received AAV-VEGF, there was almost an 80% recovery of the cardiac contractility in the infarcted region. But the thing that surprised us most was that these 2 curves started to diverge at a very early time point, 48 hours after the induction of the infarction. And this is a time which is not compatible with the formation of new blood vessels. So this data led us to think that perhaps VEGF might do something more than neoangiogenesis, and perhaps it might have some direct effect on cardiomyocytes. And to start to explore this possibility, we first look at the presence of VEGF receptors in the heart. And as you can appreciate here, whereas VEGF receptor 2 and neuropilin-1 are essentially expressed by blood vessels in the heart, VEGF receptor 1 is expressed by cardiomyocytes and is particularly localized at the intercalated disc, which are the point where adjacent cardiomyocytes get in contact. And so, to explore whether this receptor might be responsible, the activation of this receptor might be responsible of this, of the protective effect that we have observed in the dog model, we decided to test the therapeutic potential of another member of the VEGF family, which is a selective ligand for VEGF receptor 1, and this member is VEGF-B. So we used both VEGF-B and VEGF-A in a rat model of myocardial infarction, and as you can see here for this Masson 3' staining, which stain the fibrotic star in blue, VEGF-B is even more potent than VEGF-A in reducing the infarct size, and this also results in a better function of the heart, as shown here by this echocardiographic measurement, the ejection fraction, which represents the amount of blood that the heart is able to pump out at every contraction cycle, and the fractional shortening, which is another index of cardiac contractility. So again, VEGF-B is very potent in preserving both ejection fraction and fractional shortening after infarction. And the idea that he's doing this directly on cardiomyocyte is reinforced by the fact that this member of the VEGF family doesn't have any angiogenic activity. So if you look at the number of capillaries or arteries, this is not increased in the animals that have been injected with VEGF-B. And even more importantly, if you take neonatal cardiomyocytes, you put them in culture, and then you expose them to a toxic damage such as epirubicin or to reactive oxygen species, you can appreciate that both VEGF-A and mostly VEGF-B protect cardiomyocytes from apoptosis. And in addition, what these two genes do is to reactivate and to induce a gene expression program which involves several genes that control and regulate cardiac contractility, such as alpha-myosin heavy chain, the calcium pump SERCA2A, and the ryanodine receptor, as well as other genes involved in cardiac growth and hypertrophy and metabolism, such as natriuretic factors and PGC1 alpha. So the idea is that VEGF-B on one side protects the cells, the cardiomyocytes, from apoptosis, and on the other side is able to reprogram the gene expression program and to protect to increase the contractility of the cells and lead to a kind of metabolism which render the cell more resistant to an hypoxic environment. And again, we wanted to validate these results into a larger animal model, and this time we intentionally choose a model which has nothing to do with ischemia. So angiogenesis is not important in this model, and this is a model again in dog, where you can very reproducibly induce heart failure by using a pacemaker to force cardiac contraction at a very high frequency for 4 weeks. So what happens is that in 4 weeks' time, the animal almost lose all their cardiac contractility, the ejection fraction end up to be about 20%. Whereas in the animals that have received an AAV coding for VEGF-B, you can see that the ejection fraction is almost completely preserved, almost 55% of ejection fraction. And to give you an idea of what this means, if you have a patient with a 20% of ejection fraction, this is basically dead or is going to die in a very short time, whereas having 55% of ejection fraction allows you to have basically a normal life. So this is quite an important result. But if you go and read different journals of experimental cardiology, such as Circulation Research or others, you will see that over the last 10 years, hundreds or maybe thousands of molecules have been proposed as candidate genes and candidate molecules for either myocardial ischemia or heart failure. So we wanted to develop a way to understand which is the best factor and the best gene that deserve further investigation. And also starting from the consideration that VEGF, which is the prototype of angiogenic factors, eventually turn out to be very potent in protecting cardiomyocytes from cell death. Then we decided to develop a novel approach, completely unbiased a priori, which is based on the availability of libraries of thousands of genes in AAV vector, and to use these libraries to screen for novel protective genes directly in vivo in animal models of myocardial regeneration to search for novel, the most potent therapeutic candidate genes. And we call this strategy functional selection. The acronym is FunSel. So basically what we do is to generate libraries of hundreds of genes in AAV to inject this library into an animal, let's say into the heart of a mouse, then induce a damage. In this case, let's say a myocardial infarction, so something that triggers the degeneration of most of the cells. And indeed what we expect is that after this kind of damage, most of the cell will eventually die because of this degenerating stimulus. But in case some of the genes that we have injected confer a protection, the transduced cells will have a better chance to survive. And thus, if you recover the viral DNA from the cells that have survived, we should enrich our library for those genes that confer a protective phenotype in vivo. And then if you take this viral DNA and you use these genes to make a new library, you can repeat the selection until you end up with few candidate genes. They should be the best molecules that induce the survival and a better performance of your tissue directly in vivo. So for this project, we have obtained the funding to generate 2 AAV libraries. The first one corresponding to the mouse secretome, so all secreted proteins from the mouse genome, and the second one corresponding to the whole microRNA, human microRNA collection. And we are now in the process of showing the feasibility of this strategy. So we have generated several vector pools in AAV, and I can just show you one of the results we have obtained so far. So this is a PCR of the different cDNA from the secretome collection of 3 different pools. So you see that each pool has a different pattern of cDNAs. So this is the total of the 3 pools. So here you have a lot of cDNA in a single vector preparation. And what we did is to inject this AAV library into the skeletal muscle and then induce hindlimb ischemia by the ligation of the femoral artery. And after the induction of the ischemia, we harvested the survived skeletal muscle fibers, we extracted the viral DNA, and we did the PCR again. So you can see here that after the first round of selection, we cannot appreciate much differences before and after ischemia, neither in this mouse nor in the second mouse, but after the second round of selection, something started to appear here, and after the third round of selection, this band resulted to be clearly enriched after the ischemia if compared to the viral prep that we injected originally into the skeletal muscle. And so we sequenced this band and turned out to be grelin. And ghrelin is essentially known to be a hormone that controls food intake, but emerging evidence seems to indicate that it might be other peripheral effects. And indeed, we did several experiments to assess its possible therapeutic activity on both skeletal muscle and cardiomyocytes. So here again are neonatal cardiomyocytes exposed to two toxic agents, either isoproterenol or doxorubicin. And living cells are in green, dead cells are in red. So you see that both drugs act quite well in killing cardiomyocytes, whereas if you pre-treat the cardiomyocytes with an AAV vector encoded for ghrelin, these cells survive much more. And again, if you want to know more about all this part of this project, there is a poster of Giulia Ruozzi on functional selection. I just show you one final result where we tested the therapeutic potential of ghrelin in vivo, so in a mouse model, in this case, of myocardial infarction. And again, what we observe here is this is an echocardiography movie of an infarcted heart. So this is the left ventricle, the main ventricle of the heart. And you see here that there is a part of the ventricle which is missing, is not contracting. So this is the myocardial infarction. Whereas in the case of a mouse injected with ghrelin, the infarction is much smaller, and you can appreciate here that the heart is contracted much better than the previous one. So believe me, this heart is contracting almost as all our hearts. So the cardiac function is completely preserved in this mouse. So as a summary, I will just finish with 3 sentences that summarize what I show you. So we can obtain a potent angiogenic activity exploiting AAV vectors to express angiogenic factors such as VEGF-A. Then we have seen a striking protecting effect of VEGF-B on cardiac function by a direct binding of VEGF receptor 1 exposed on cardiomyocytes. And last, I show you the FunSel, so our novel strategy to select cardioprotective genes through in vivo gene library selection, which has already identified ghrelin as a cardioprotective factor. Just a final slide to acknowledge the work of the people involved in all the experiments I showed you before. Mostly Alessandro Carrer and Silvia Moimas for all the angiogenesis story. Lorena Zentilin for all the VEGF-B work, but also as she is leading our AAV vector facility at ICGEB. And Giulia Ruozzi, Antonella Falcione, Francesca Bortolotti, together with Matteo Del Ferro and Fabrizio Pirozzi, they are actively working on the functional selection project. And of course, Mauro, who is sharing with us all of this and much more, and all the external collaborators. So, thank you very much.